Hello, BISC130. This is the beginning of recorded lecture 4.1. We are still in the chapter on DNA structure and function, but we're talking about some other things. We're going to start by talking about DNA repair, but to get here, I actually want to bring this up again. So I know this was part of the previous lecture, the previous exam. I'm bringing up all this madness again just to point out that this entire process of replicating DNA and creating DNA that's complementary to the original DNA and the template here, that this process is not perfect. You can get mismatches. You can get mistakes during the process of DNA replication. And here we can see, yep, one of these is leading, one of these is lagging. The 5' 3' are not labeled here, but yeah, we got DNA daughter strands, C to G, A to T, oopsie, G paired with T. So this kind of mismatching or mispairing, same thing, this can happen. And as this is happening, the machinery of DNA replication is looking for these mistakes, and it's trying to fix them when they happen. But even so, you sometimes get these oopsie doodles making it past the proofreading mechanisms, and sometimes you get mispairings in your final product. So, to summarize, DNA polymerase 1 or 3, any of these, they proofread DNA as it is being made. They are checking for mispairings, but they are not perfect. So we could try to fix these mispairings after replication, but even that is not perfect. Sometimes we get mispairings or mismatches in the final product. So related to this is the idea of damage to DNA. So there are a lot of different ways to mess DNA up. Chemicals, and again, this is a very broad category because there are many chemicals out there, but things like asbestos or industrial chemicals like ethidium bromide or any of the chemical components of cigarette smoke. There are tons of different chemicals that could cause damage to DNA in a lot of different ways. We can try to avoid these, obviously, but it's hard to avoid ultraviolet light. If you're exposed to sunlight, you're exposed to some amount of ultraviolet light. You could limit your exposure by, wearing a hat, putting on sunscreen, but ultraviolet light does cause DNA damage. And this third one I'm listing here is completely unavoidable. Things called oxygen-free radicals are chemical compounds that are simply a byproduct of metabolism. So the metabolism your cells are doing creates some amount of these oxygen-free radicals, which damage DNA. So you can try to limit your exposure, but at the end of the day, your DNA is getting damaged no matter what. We're going to need ways to fix it. So before we talk about fixing, I also want to point out, just like there are many different ways to damage DNA, there are many different types of DNA damage. Sometimes this could be individual nucleotides getting screwed up. So here's a guanine and it's had an extra chemical group added to it. Now it's a slightly different guanine, and you may not think that this is going to be a big deal, just this little extra methyl group here or whatever. But yeah, just modifying the nitrogenous bases or the nucleotides can really mess things up as far as DNA goes. So sometimes damage is small scale like this. Sometimes it's bigger. Oh yeah, a crude rendition of a sun here. Ultraviolet light does indeed cause this type of DNA damage where you link a couple of adjacent thymine bases to one another, and that's causing the DNA to bulge out. Don't sweat either of these details. They're just making this point. There are lots of different ways in which DNA can be damaged. The main point of this section, though, is that the problem with DNA damage comes in replication. If damaged DNA, regardless of what type it is or what caused it, damaged DNA will not pair correctly, will not find its correct partner, A to T, C to G, will not pair correctly during DNA replication. Let's walk through what this can look like. So here's a sequence of DNA that I made up. Again, you do not have to memorize this exact sequence, but follow through on this particular example. So here's our sequence of DNA. Again, it's double-stranded. So yeah, CTA, GAAC. The other strand is GAT, C, T, T, G. You know, A's to T's, C's to G's. Let's mess this up a bit. Let's do some of this. Let's create some 6-O-methyl-guanine. Let's pull a star on this G. This guanine has been damaged. Now, let's go through the process of DNA replication. We know that both of these strands are going to be replicated. One's going to be leading, one's going to be lagging, whatever. Let's just focus on the top strand and how it gets replicated. So C is going to pair with G, T pairs with A pairs with T. But here's the messed up guanine. And the machinery that's doing DNA replication is going to look at this messed up guanine, and it may not recognize it as guanine. There's a very good chance that the DNA replication machinery is going to look at this messed up guanine and think that it's an adenine, putting a thymine across from it. And then the rest is going to be normal, you know, A's to T's, A's to T's, C's to G's, and there we go. At this point, we can still try to fix things. But what becomes truly catastrophic is when we do a second replication. And this may seem like too much, but in the cells that are actively dividing in your body, they're doing DNA replication and then finishing mitosis and then doing another DNA replication as, you know, as part of the cell cycle, and then they'll do mitosis and cytokinesis. And so, yeah, it's not that unusual for a cell to do DNA replication and then turn around and do it again. So let's replicate this DNA again. We already know what happens when we try to replicate this top strand, so let's do a second DNA replication, but let's just focus on this bottom strand here, the GATTTTG. So let's replicate this strand. So G is, or you're going to put a C across from the G and a T across from the A, and you know A's adenines partner against thymines, and yep, there's C to G, and yeah, we finished replication. Where's the damage? Where's the messed up nucleotide? Where's the error? If we wanted to fix things, what here needs to be fixed? Well, we know that this particular pairing, AT, is not what we started with. Originally, this position was a GC pair. But AT, this is a perfectly normal adenine, this is a perfectly normal thymine, and this is correctly partnered up. This doesn't look wrong, like to the cell. This doesn't look wrong at all. There is no way to fix this. This, if I can scroll back a bit, this was damaged DNA. The cell can recognize that this guanine is not right, and there are ways to try to fix this damage that we'll talk about in just a minute. But after you've gone through a couple of replications, and you have something like this, there is no way to fix this. This is what we call a mutation. And again, mutation and damage are two terms that are very similar to one another, but they are not exactly the same. The biggest difference is that you can fix DNA damage. You cannot fix a mutation. A mutation is, actually, it's in the key terms. So yeah, damaged DNA or unrepaired mismatches. This is why I brought up mismatches at the very beginning of this lecture. We could pretend that instead of, let's go and scroll back here. Instead of, a messed up G being partnered with a T, this was a perfectly normal G partnered up with a T, just as part of a mismatch that would have led to the exact same scenario happening. So whether it's damaged DNA or an unrepaired mismatch, these things can lead to mutations. To read from the key terms, a mutation is a stable change in the nucleotide sequence of a genome. Stable change being an important part of that. Mutations cannot be repaired. It's just a different sequence now, and there's no way for the cell to know that this is different than what it was a couple of replications ago. Now, the definition of mutation was not a negative one. It just says stable change. And not all mutations are necessarily bad. It's just different. It's just a change. That being said, it's usually bad. If you're taking a very complicated, sequence for a very specific protein and you're just swapping things around, it's usually going to mess things up. I mean, imagine you had a blueprint for a very complicated house and you just erase one of the walls and put in a different structural element like a toilet instead. It's probably going to have some issues. So in general, mutations are things that we want to avoid because these stable changes usually have a negative effect on the product of that DNA, of that gene. So we want to prevent mutations from happening because, again, there's no way to fix these. We want to prevent mutations by repairing DNA damage before replication. And this concept should sound familiar to you. We saw this in all the cell cycle, or as part of cell cycle regulation from a few chapters ago. We had all of these lovely proteins where we were talking about these checkpoints, especially this one, before DNA replication. You want to check DNA, make sure it's fine before you go through replication. And, this is why, now that we know a little bit more about DNA and how it works, we're coming back and closing the loop here. Now we understand why this checkpoint was so important. We want to prevent mutations from happening in the first place by repairing DNA damage before you get to replication. So now let's actually talk about DNA repair. So I want to start by saying just like there are many different types of DNA damage, there are many different DNA repair mechanisms that exist. I just want to talk about one of these in a little detail, something called nucleotide excision repair. So let's walk through this process. Here we've got normal looking DNA. Step one, DNA is damaged. And again, it's recognizable at this point as damage. These nucleotides are not partnering up correctly with one another. I think this particular example is trying to show something like this caused by ultraviolet light, but really this repair mechanism I'm about to talk about can fix any sort of small scale DNA damage. So you have some sort of damage that occurs. The enzyme that's going to fix this damage is going to come in. As always, this is a big, complicated, very fascinating protein, with multiple parts to it and a horrendously complicated three-dimensional structure. It's just a purple oval. This enzyme called the UVRABC complex. Technically, it's three separate proteins: the UVRA protein, the UVRB protein, and the UVRC protein coming together in a quaternary structure, if you remember that term from many chapters ago, to form what's called the UVRABC complex. It's going to come in and it's going to cut out some DNA. And a very important thing to note is that it's not just cutting out the damaged nucleotide or two nucleotides, it's cutting several nucleotides around that damage. You want to make sure that you cut out and excise a large chunk of DNA, not just rip out one or two bad nucleotides. You want there to be a very visible, recognizable gap in the DNA. Why do you want there to be a big gap? Well, you want there to be a big gap so that we could fill it in. So we need some sort of enzyme that could recognize this small little gap and fill it in with freshly made DNA. This functionality may sound familiar to you. To call back to, again, last exam, but previous lecture, we saw this functionality in DNA polymerase 1. It removed the RNA primers, creating a short little gap, and it filled in that gap with DNA. So here, this has nothing to do with DNA replication. This is a repair mechanism, but we need something that does the same job of DNA polymerase 1. Let's just use DNA polymerase 1. There's no need to invent some whole new enzyme. This can serve multiple purposes. So yeah, it comes in, it fills in this gap with freshly made DNA. There we've got our DNA, but it looks like we're missing a little piece here. It looks like one little phosphodiester bond is not quite filled in yet. And you know what? We saw this problem before as well. And guess what fixed that problem before? It was DNA ligase. And so once again, not having to reinvent some new enzyme, we're going to use DNA ligase to seal this together. And now we've got big, freshly made DNA, and yeah, looks just like it did before it was damaged. So again, I like this figure, but let me summarize this. This is called nucleotide excision repair. The UVRABC complex recognizes the damage, cuts out a damaged area, again, underlining area and pointing out large chunk. It's not just removing one nucleotide, cutting out a large area of DNA, and then DNA polymerase 1 fills in the gap, DNA ligase seals it all back together. So again, there are lots of different repair mechanisms to deal with lots of different types of damage. This is just the only one that I wanted to walk through and talk about. Now, the last topic in this chapter is actually types of mutations. This is a very interesting and very important topic to discuss. However, I'm going to save this for later. Trust me, I really think that this will make sense after we've talked about some of the things in the next chapter. So yeah, I'm moving on to chapter 15, genes and proteins. Don't worry, we will circle back to this in its own time. But yeah, we're going to randomly have some chapter 14 section in the middle of chapter 15. But yeah, trust me, it'll make more sense after we do some of this stuff. So, Chapter 15, genes and proteins. I mean, it's, very similar to what we've been talking about, DNA structure and function. But now we're going to be talking about how this, in this chapter, how this DNA, how this sequence of GATTCAG, whatever, actually does stuff. So we're going to start this chapter with something called, it sounds way more overblown than it actually is. We're going to start by talking about something called the central dogma of molecular biology. And again, it sounds very grand. Trust me, it's not as intimidating as it sounds. The central dogma of molecular biology is going to describe the flow of information in cells. And once again, just this sounds really complicated. It's not. The central dogma, DNA, RNA, protein. That's it. That's the thing. That's the central dogma. This is how information flows in cells. Information, instructions are stored in the DNA, they're copied over into the RNA, and then they are used to build proteins. If you want this a little bit more visually, DNA to RNA to protein, that's central dogma. Now, we do have, a little bit of a circle here because DNA can replicate itself. And there are names for these two steps. So as we use the information in DNA to build RNA, that process is called transcription. And as we use the information in RNA to build protein, that process is called translation. I have it on this slide as well. DNA to RNA is called transcription. RNA to protein is called translation. Side story. So, we've seen this before. These are two words that look very similar to one another and will be very easy to mix up on a multiple choice question. Hint, hint. Or perhaps many multiple choice questions. We're going to talk a lot about transcription. We're going to talk a lot about the details of translation. It's really important to know which one is which and not get these two terms mixed up. There's a reason why these things have the names that they have. So transcription comes from the word transcript. A transcript is a copy of something. So yeah, you've got, your grades stored, in a computer somewhere. I guess back in the day it used to be written on a piece of paper at the registrar's office. You've got your grades written down in a transcript. If you want a copy of that sent over to, some other university or a job you're applying to or, something like that, the transcript is a copy of that document sent to someone else. If you wrote down some of these notes from this lecture and your friend didn't want to watch this lecture themselves, they just wanted a copy of the notes, they would make a transcript of your notes to get all that same information. This process of going from DNA to RNA is called transcription because the RNA really is just a copy of DNA. It's DNA and RNA are the same basic thing. They're polymers of nucleotides. Yeah, RNA is a little bit different. It's got uracil instead of thymine. So if your friend hand copies your notes, maybe they have a blue pen instead of a black pen. But at the end of the day, it's the same basic type of thing. Polymer of nucleotides, it's just a copy of it. So that's why transcription is called transcription. It's a copy. Translation comes from the root word translate, which, is going from one language to another language. If you've got some Spanish homework where you have to translate a chapter of a book from Spanish to English or English to Spanish. And yeah, RNA and protein, these are completely different languages. So RNA is a polymer of nucleotides. DNA is a polymer of nucleotides. But protein, that's a polymer of amino acids. Completely different type of thing. So the word translation makes a lot of sense here. So I'm not writing any of this down. This was just my, sort of side note story to help not get these two terms mixed up with one another. The other thing I want to point out here is this is the flow of information. When I say DNA goes to RNA, we are not taking a DNA double helix and physically morphing it to become RNA. We are not transforming this molecule into this molecule. This is the flow of information. We are using the information in this molecule as the instructions for how to build this molecule. And the same thing here. We're not morphing RNA into protein. We're not reconstructing or reconfiguring RNA into protein. We are taking the information in RNA and using that as the instructions for how to build this protein. You don't take the blueprint and do some fancy origami and build the house out of the blueprint. You use the blueprint as the instructions for how to construct the house. So a lot of students do get this mixed up just because the arrows sort of imply that. But no, this is information that's moving from one to the next. Okay. Finally, we can move on to prokaryotic transcription. Okay, so yes, once again, I will highlight the differences between prokaryotes and eukaryotes when it comes to transcription. We're going to start by talking about prokaryotes. Lucky for us, eukaryotes are going to be very similar. Technically, I'm talking about prokaryotes to start, and then I will highlight how eukaryotes are different when we finish talking about prokaryotes. So, okay, this whole process of transcription is carried out by a single enzyme called RNA polymerase. Very similar to DNA polymerase. Remember, DNA polymerase built DNA. Well, RNA polymerase is going to build RNA. It makes sense. And again, my figures are going to, show RNA polymerase as a little oval blob or whatever. But nope, it's a super complicated protein with a massive quaternary structure and, whatever. But yeah, this is RNA polymerase. So we could talk about the process of transcription as carried out by RNA polymerase as three distinct phases. The first phase is called initiation. Initiation begins when the RNA polymerase, here's the orangish tan blob, where the RNA polymerase binds to the DNA. Not just anywhere on the DNA. The RNA polymerase is going to bind to a specific region of the DNA called the promoter or promoter region. The promoter is right before where you're going to start transcription. The gene that you are transcribing is going to be right after this promoter. The whole job of the promoter is to tell RNA polymerase where it's supposed to bind so it can start things. You don't want to start this gene in the middle. You want to start, 3/4 of the way through. You want to start at the beginning. And the only way RNA Pol knows where the beginning is if it has some sort of indication of that, and that's the promoter. You'll notice in this figure, the TTGA, CG, and whatever, Don't memorize these exact sequences, but it is important to note that the promoter does have a very specific sequence. This RNA polymerase knows what a promoter is and where the promoter is and how to bind to it because of the sequence that the promoter has. If the promoter did not have the correct sequence, RNA polymerase would not be binding to it. So, important part of this. So yes, initiation. RNA polymerase binds to the promoter. Promoter is defined in the key terms as a region of DNA with a specific sequence. Again, not a very long definition, but it is a very specific sequence. Importantly, the promoter is upstream or right before where transcription will begin. Now, in some ways, we can compare the process of transcription to the process of DNA replication. In DNA replication, we needed to create a single-stranded DNA template so we could build DNA complementary to this single-stranded DNA, and we used helicase to do that. We're doing a similar thing here in transcription. We need single-stranded DNA in order to build RNA complementary to it. The difference is we are not going to be transcribing the entire chromosome. So we do not need to unzip the entire chromosome like we did with DNA replication. Some unzipping needs to occur, but not this sort of large-scale thing. The unzipping is going to happen in what is called a transcription bubble. So here this is a good figure to summarize the whole process of transcription. Here we've got our DNA double helix. Here's RNA polymerase, the orange bean, or the purple bean right here. And yeah, you can see in this region right here, it has been unzipped. There is single-stranded DNA, and there is single-stranded DNA. This little bubble where it's been unzipped is called, appropriately, the transcription bubble. So we're still in initiation. RNA pol binds to the promoter, and it forms the transcription bubble. The key terms define a transcription bubble as a region of locally unwound DNA that allows for transcription. Again, it's unwound, it's unzipped, it's single-stranded, but it's just this local region right here. Okay, now that we've started things with initiation, our second phase is called elongation. This is where we're going to actually start building RNA. The way that RNA polymerase is going to build RNA is in the 5' to 3' direction. Oh no, not this again. So yes, getting flashbacks to last chapter, last exam maybe. This enzyme, RNA polymerase, has the same limitation that DNA polymerase had in that it is only able to construct its thing in the 5' to 3' direction. We can see this here. We've labeled the strands of DNA, the RNA, and yes, this arrow is showing the site of unwind, or the direction of unwinding, the direction of this transcription bubble. The promoter must have been up here somewhere. The bubble is moving along, and here is the RNA being constructed 5' to 3' in that direction. Now, in the last chapter, this became a nightmare for us because of the leading strand, lagging strand stuff. Luckily for us, if we look at this figure, essentially this is just quote-unquote leading strand, 5' to 3' toward the site of unwinding, toward where the transcription bubble is moving. There is another template available here. There's more single-stranded DNA available here. It is not transcribed, so there is no lagging strand. In DNA replication, we had to replicate both. In transcription, we're just transcribing one of these. So phew, crisis averted. This turns out not to be that complicated, but it is still important to note that it is building in that direction. Now, there are two very important terms when it comes to talking about the DNA as it is being read by RNA polymerase to build RNA. You can see them labeled here, coding strand and template strand, but what do those mean? Well, the RNA sequence that is being built is complementary to the template strand of DNA. So real quick side note, because we are building an RNA-DNA hybrid here. If you look here, this is RNA, DNA. This is double-stranded DNA, RNA. They're hydrogen bonding with one another. Double-stranded DNA, RNA hybrid. The rules of base pairing when it comes to DNA base pairing with RNA is a C in the DNA will pair with a G in the RNA. Okay, that's exactly like it was with DNA. A G in the DNA will pair with a C in the RNA. Okay, that's familiar. A T in the DNA pairs with an A in the RNA, and an A in the DNA pairs with, not a T in the RNA. Remember, RNA does not have thymine, it has uracil instead. An A in the DNA pairs with a U in the RNA. So this is the one little difference between the rules of base pairing we learned for DNA to DNA in the last chapter. Again, I know I introduced this way earlier in the quarter, but I'm bringing it up again. RNA uses uracil instead of thymine, so as we're constructing this RNA, it's going to partner up with the DNA according to these rules of complementarity. So, like I said, the RNA sequence that we build is going to be complementary to the template strand of DNA. Here is the template strand, the one down here, and you can see it's being built complementary to this. The other strand is called the coding strand. And the RNA sequence that we are going to build, the piece of RNA that we are building, is going to be the same as the coding strand. Now, these two statements might be a little confusing, so I want to give an example. Do not memorize these exact sequences. They're just examples. Let's say we have a coding strand up here, and this is the sequence. A, T, G, C, T, A, G. Whatever, I just made it up. We know what the template strand sequence is going to be like. The template strand is complementary to the coding strand. These are just two strands of DNA. DNA is always complementary to itself in its two strands. A pairs with T, T pairs with A, C, G, A, T, T, C. Easy. It's DNA to DNA. So what about the RNA? Well, as I said, the RNA sequence that we build is going to be complementary to the template strand. So reading the template, again, it's called a template. It's what we read. It's what's used. T in the template is going to pair with A in the RNA. A in the template is going to partner with U in the RNA. C to G, G to C, A to U, T to A, T to A, C to G. This is what I mean when I say the RNA sequence that we build is complementary to the template strand of DNA. But what about the other statement here? The RNA sequence is the same as the coding strand. Remember, the coding strand is the one that we did not read off of. The coding strand is the one that's just sitting here, not being touched at all. Well, let's compare the RNA sequence to the coding strand sequence. They're not complementary, they are the same. A, the same. U, T, well, okay, they're not exactly the same. Remember, DNA uses thymine and RNA uses uracil instead, but they are equivalent, they are the same. G, C, U, T, again, that thing again, A, G. It's the same sequence. The T's and U's are different, but other than that, the coding strand is the same as the RNA strand. So, again, this is a great example to return to if you get lost in these statements. I'm trying to make these as as pithy and succinct as I can. But these are the two important statements here to understand what these two terms mean: coding strand and template strand. The RNA sequence is complementary to the template, and the RNA sequence is the same as the coding strand of DNA. Okay. Now, the other thing that's happening here is as we are building RNA complementary to the template, the transcription bubble is moving along. It is going to unzip more of this DNA, and it's going to zip back up again as the transcription bubble sort of passes by. Elongation. The DNA rewinds as the transcription bubble passes by. And this is going to continue on until we make it to our third phase, called termination, where things are going to end. There are a couple of different ways that this can happen. Let's start by talking about something called row-dependent termination. So I didn't show it earlier because I was trying to keep things simple, but in a lot of transcription, there's an extra protein called row factor that binds to the RNA early on, and it moves along in the same direction as the transcription bubble. The row factor is sort of like chasing down the RNA polymerase, chasing down this transcription bubble. But it's not gonna catch the RNA polymerase because they're moving at about the same speed. However, if this RNA polymerase hits a bunch of G's in a row, it's just something about guanine in specific and having a bunch of those, at the same time, G, hitting a bunch of G's all together is going to cause RNA polymerase to stall a little bit, to slow down a little bit. That is going to allow the row factor protein to catch up to it, and that is going to terminate the whole thing. It's going to cause the RNA to be released. It's going to cause the RNA polymerase to release. The DNA is going to zip back up again. Everything's going to be done. You're finished. And it always terminates at the same place because this is going to happen right after RNA pol hits a G sequence in the DNA. So to summarize, in termination, the row protein follows behind the transcription bubble. At a sequence of many G's in a row, the RNA polymerase stalls, row catches up to it, and ends transcription. I mentioned there were two common ways in which transcription can be ended. Row was just one of these. The other way that transcription can end is what is called row independent termination. This does not involve the row protein. This involves something called an RNA hairpin. So we're talking about, if you don't have long hair, maybe you're not familiar with these. This is a type of hairpin. This is named after this hair accessory because this is RNA and then a little loop and then more RNA. So RNA is not normally double-stranded. But if you have a sequence of RNA that is C, G, 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 C, G, and then after a short while later, a sequence of RNA that is exactly complementary to that, RNA will form a double-stranded structure if it has this kind of opportunity. And as it turns out, the formation of this kind of hairpin in the RNA is enough to yank the RNA out of, RNA polymerase is not shown here, but we see the little transcription bubble. So RNA polymerase would be here building this RNA. The creation of this RNA hairpin is going to be enough to yank this out of RNA polymerase and cause the whole thing to terminate and everything releases and you have your RNA. So still under termination, a hairpin structure forms in the RNA that causes transcription to end. That's all I want to say about the row independent. You get a hairpin in the RNA, that makes transcription stop. Either one of these, row or the hairpin, are ways that we can cause termination during transcription. What about us? What about eukaryotes? I told you I would do this compare and contrast because technically all of this was prokaryotes. So what about eukaryotic transcription? Luckily for us, it's fundamentally the same as pro. You have the same three stages. We've got a promoter. We've got a transcription bubble. The basics of everything I just talked about are the same. There are a few small differences though. One of them is that eukaryotes, more complicated as always, actually have three different versions of the RNA polymerase enzyme. RNA pol 1, RNA pol 2, and RNA pol 3. Don't memorize this table, but it's just kind of showing you that these different polymerases build different types of things and are active in different parts of the cell, and just have slightly different jobs. Prokaryotes only have a single RNA polymerase, but eukaryotes, we've got different ones that do different things. So fundamentally the same, but as I list the differences, 3 different RNA polymerases, 4 different types of RNA. The other major difference has to do with processing. So here's an example of a sort of raw transcript from a eukaryote. You have the beginning, the middle, the end. Within this piece of RNA, there are a couple of different types of region. There are exons and introns. So this is the raw product of transcription. This is what we build straight, fresh out of RNA polymerase. Before this transcript is ready to go on to translation and do its job, to be a blueprint, to tell the cell how to build a protein, you need to cut out the introns, remove the introns, and glue together the exons. You'll notice how, this is our raw transcript, this is our final product. The introns have been removed, the exons have been put together. So, and again, this is a eukaryote thing. Prokaryotes don't do this nonsense, only we do. Introns are spliced out and exons are kept in. Introns are defined in the key terms as an intervening sequence. That's what int is, supposed to be short for, intervening. You could think of it as in the way, because ultimately these things need to be removed. Introns, intervening sequence that is spliced out of RNA during processing. That's what the key terms say. And exons are kept in. The key terms definition of exon is an expressed sequence. That's what the X is supposed to be short for, expressed. An expressed sequence that is kept in the RNA after processing. So introns removed, exons kept in. Why, though? This seems like a tremendous and stupid waste. Why do the introns even exist in the DNA if you're just going to remove them? What's the purpose? The answer to why these things exist is, I think, unfortunately, or maybe fortunately, well outside the scope of an introductory biology class. There are fascinating examples of introns, being left in, and some of them being taken out in order to get multiple different RNAs out of a single starting RNA. There are examples of changing the order of exons in how they're spliced back together in order to get multiple proteins out of a single gene. None of that is worth going into in this course. I'm sorry that I'm not really answering the question of why this happens, but for here, you need to know that this is something eukaryotes do that prokaryotes do not do. We've got introns that we remove, we've got exons that we keep in. There's one last difference between eukaryotic transcription and prokaryotic transcription, and that has to do with the size of these cells. So this is approximately to scale. The prokaryotic cell is pretty small. So if you build an RNA transcript, for that transcript to get where it needs to go in the cell is pretty easy. It doesn't have to travel very far. However, in eukaryotes, transcription is taking place in the nucleus, and that piece of RNA needs to go, lots of different places. Maybe it needs to go here to the rough ER. Maybe it needs to go out here to the cytoplasm. It has to travel across the cell, across cell membranes sometimes, and it has to make quite a lengthy journey. So because of this lengthy journey that eukaryotic transcripts need to make, our transcripts need to be processed. So here's our exon, and we add some extra things to the 5' end and some extra stuff to the 3' end to just extend its shelf life, so it can make it to where it needs to go, and to mark it for transport so it can be sent to where it needs to go. So let me summarize this. RNA, again, this is a eukaryote thing. RNA is processed in order to extend stability and to facilitate export from the nucleus, to get it out of the nucleus where it's made to wherever it needs to go, and to have the shelf life to do that. So again, the fundamentals are the same between prokaryotes and eukaryotes, but we have multiple RNA pols, we've got introns, exons, we've got processing of these RNAs. Other than that, it's pretty much the same between eukaryotes and prokaryotes. Okay, we have finished talking about transcription. Before we move on to translation, there are a few more things to talk about, but I'll have to talk about those in the next recorded lecture. So this is typically where I finish up with things for this one. This is the end of recorded lecture 4.1. We'll talk more about genes and proteins and finally get to translation in the next one.